Welcome to the Pharmacad. So today we are going to discuss the question paper of Pharmaceutical Organic Chemistry 3, Semester 4. So we will solve one question paper in this lecture. So let's start with the first question. So the first question is, identify the relationship between the molecule X and Y. So see, this is in the form of Fischer formula, but the second structure is in the Newman projection. So first we have to convert this into Fischer. So always remember that when the Newman is given and when we have to convert it into Fischer, first we have to rotate it in 180 degree. So when we will rotate this into 180 degree, okay, so how we will get the structure? So the CnH2 with this hydrogen, with this chlorine will remain as it is. Okay. So when we are going to rotate the rarer carbon, which is with this bromine, this methyl and this chlorine with 180 degree. So this chlorine will displace from this position to this position. Okay, so we will get chlorine over here, whereas methyl over here and bromine over here. Okay, after rotation by 180 degree. Now we have to draw the Fischer projection in this manner. So how we will get the structure? So the CONH2 will remain over here, whereas this methyl group over here, this carbon, the frontal carbon is with hydrogen and chlorine, and the rarer one is with bromine and chlorine. Okay, now we have to check the relationship in between this and this compound. So as you can see, the difference is at this carbon as well as this carbon, because in first structure in X, we are having chlorine is on left hand side, whereas in case of Y, we are having chlorine at right hand side. Talking about the second carbon, the chlorine is on right hand side. And in this case, we are having chlorine is on right hand side. It means the change in the position of atom only at single position, this carbon. So you have to remember, whenever there is change at single position, the relationship in between those compounds is diastereomer. If we are having change at two positions, in that case, the relationship will be enantiomer. So the correct answer of this MCQ is diastereomers. The second MCQ is, which of the following molecule is chiral? Now, how we can identify whether the compound is chiral or not? So for that, first we have to identify whether that compound is having asymmetric carbon or not. So how we can identify what is asymmetric carbon? So the carbon which is with four different atoms or groups. If it is with four different atoms or groups, then only we can call it as asymmetric carbon. And whenever we are having that carbon in our compound, that compound become chiral molecule. So we will see in case of first molecule. So you can see this carbon is with methyl, it is with hydroxy, it is with CH2, 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 whereas the another one is again CH2 and CH2. It means what? We are having two same groups over here. So this is not chiral, this is achiral. In case of second molecule, if we are considering this carbon atom is with one NH2 amino group. It is with one methyl group. The whole group is as a substituent, and this carbon is having one hydrogen over there. It means what? This carbon is with 4 different atoms or groups. That is nothing but the asymmetric carbon. That is nothing but the chiral carbon. And the denotation is with the help of this asterisk mark. So the second compound is chiral. So the answer is option B. Next question, nomenclate the given molecule. Now this molecule is having total 6 carbon with aldehyde and with alcohol. So I hope you know the structure of glucose. This is nothing but the glucose. Now we have to identify whether the carbon is having R or S configuration. Before that, we have to check the numbering pattern. So as we all know that the aldehyde carbon is having first carbon atom, the second one is with this hydroxy, the third carbon is this, fourth one, and fifth one, and sixth one. So you can see for second position, they have given the same configuration, that is R and R. We will check for third carbon atom, that is this carbon atom. So we will take that carbon atom separately. That carbon is with this hydrogen. It is with hydroxy. It is with carbon with hydroxy, and that carbon is with aldehyde group. Whereas another substitution is this whole group, that is with CHOH, again CHOH, and then CH2OH. Okay, so we have to give the numbering as per the atomic number. So you can see this carbon is directly attached with oxygen. That's why the priority will go to the hydroxy group. So this becomes first. The second one, this one or this one. So see, in this case of carbon, the another carbon is directly attached to the double bonded oxygen, whereas this carbon is attached with the single bonded oxygen. That's why the priority will go towards this group. So this will get the second priority, whereas this whole group will get the third priority, and this hydrogen will get the fourth priority, that is the least priority. Okay, it means what? We are having fourth over here, third over here, first over here, and second over here. Now we have to check the rotation from 1 to 2 to 3, and the fourth group is negligible. Okay, so when we will rotate in this manner, this is in the manner of clockwise. But there is one rule, if you remember, Kahn and Gold rule says that the least priority group should be on vertical line. If it is not on vertical line, then the configuration will change from R to S or S to R, vice versa. So in this case, our rotation is in the form of clockwise, but the fourth group is not on vertical line. That's why the configuration become S. It means what? The third carbon is having S configuration. So we can conclude our answer, that is option B. Okay. I hope this MCQ is clear for all of you. Now the next question is a mixture of equal quantities of 2 enantiomer. So 2 enantiomer, it means what? Dextro or levo, or R or S. 2 equal quantities. It means 50% R plus 50% S. Okay, or 50% dextro plus 50% levo, which is equals to the optically inactive mixture, and that mixture is nothing but the racemic mixture. Remember this thing. So the correct answer is option C. Okay. Next MCQ is, which of the following compound has R configuration? Okay, so over here again we have to check whether the rotation is in the clockwise manner or anti-clockwise manner. So for first compound, the priority will go towards the chlorine. As per the atomic number, we have to give the priority. So chlorine will get the first priority, so it becomes 1. Whereas this carbon with oxygen becomes 2, this methyl becomes 3, and the hydrogen has the least priority, so 4. So we have to rotate from 1 to 2 and 2 to 3. It means in this manner. Okay, this fourth one is negligible. So this rotation is in the form of anti-clockwise, whereas the least priority group, that is hydrogen, is on solid wage. But that hydrogen should be on dash wage. If it is on solid wage, so the R becomes S and S become R. The rotation is in the form of anti-clockwise. It means it should be S. But the least priority atom is on solid wage. That's why it becomes R. So the configuration of this is R configuration. Whereas in case of second compound, see, first priority, then second, then third, and then fourth. 1 to 2 and 2 to 3, it means anti-clockwise. The hydrogen least priority group is on dash weight. That's why the configuration is S. Whereas in case of third compound, first, second, it is having two same atoms. So it is optically inactive compound. In case of compound 4, first, second, third, and fourth. So fourth is negligible, so it is in the form of anti-clockwise. H is on vertical line, so the configuration remains as it is, that is S. So the correct answer, they are asking R configuration, which is of compound 1. So the answer is option A. Okay. Now the next MCQ, identify the correct name with configuration of the following compound. So first we have to think on the numbering pattern of this alkene. So as we all know that the priority towards the acid and the numbering will start from this manner: 1, 2, 3, 4, and 5. This is the longest straight chain on which we are having substitution of this methyl and this bromine, which is on third and fourth position. So we have to check the options in which we are having 4 bromo and 3 methyl. So 4 bromo and 3 methyl, we are having two options over here, A and B. The only difference is in the manner of Z and E, whether it is in same manner or it is on the opposite plane, we have to check. So how we can check? First, we have to check the highest priority group. So when we are comparing in this methyl and this one CH2COOH, which is the substituent of this carbon atom, so the highest priority group is on this side. When we are comparing for this carbon atom, the highest group is on this side. It means what? The highest priority groups are on opposite side. So remember, whenever they are on same side, it is Z. Whenever they are on opposite side, it is E. So the answer is E, 4-bromo 3-methyl pent 3-enoic acid. Why 3-enoic acid? Because on third position, we are having carbon-carbon double bond. I hope this is clear for all of you. Next question is, which of the following decrease in order of stability of cyclohexane conformation is correct? So we know that the cyclohexane has total 4 conformations, that is chair, twist boat and half chair. Okay, the chair conformation is having the highest stability because the all angles are 109 degree 28 minutes. Okay, so if we have to compare in between these conformation, so the highest stability conformation is with chair conformation. So the first is chair, okay, which is having 0 energy. After this, the twist board, because it is having 5.5 kilocalorie per mole energy is required to form this twist conformation. After this, boat conformation, because it requires 6.5 kilocalorie per mole. And after boat, there is half chair, because half chair requires 10 kilocalorie per mole energy. So as we know that the relationship in between energy and stability is inversely proportional. If the energy is high, the stability is low. Okay, so on this basis, we can conclude the option, that is option A, chair, twist boat, and then half chair. Okay, next MCQ is, which are stereoisomers resulting from hindered rotation about single bonds, where the steric strain barrier to the rotation is high enough. Now, when steric strain rotation is possible? When we are having two bulky groups over there, okay, on the opposite side of that single bond. And if you remember, whenever we are having two phenyl rings, okay, with the single bond, so these two phenyl rings are too much bulky groups. And because of that bulky group, there will be hindrance in the rotation about this single bond. And that is nothing but the atrop isomers. Remember this thing. So the answer is option D. Next question. Identify the wrong statement about geometrical isomerism. So as we know that geometrical isomerism is possible whenever we are having carbon-carbon double bond. Then only the geometrical isomerism is possible. Okay, so the first statement is it must contain carbon-carbon double bond. So this statement is correct. Second thing, two different atoms or group must be linked to each doubly bonded carbon atom. It means what? If this carbon is with A, then the another substituent should be B. It should not be A. See, if this carbon is having A and B substitution and this carbon is having A and B, that is fine. But the same carbon should not have the same substituent. Okay, so this statement is also true. Next is, cis and trans are geometrical isomerism. Yes. Cis, it means when the highest priority groups are on same side, then it is cis. If both of them are on opposite side, then it becomes trans. So this statement is also true. It occurs due to the rotation of carbon-carbon single bond. No, it is when we are having carbon-carbon double bond. So this statement is false. Okay, they are asking wrong statement. That is option D. Next question, nomenclate the following structure. So this is two heterocycles which are fused with each other. So out of that, the first heterocycle is with oxygen and nitrogen, whereas the second heterocycle is with nitrogen and nitrogen, which is on first and second position. So see, whenever we are having oxygen as per the priority, the heterocycle ring, which is with oxygen and nitrogen, will get the priority. So that becomes our parent ring. Okay, so first we have to give the numbering. Over here we are having nitrogen with this NH with this nitrogen, and here we are having double bond and double bond. So first we will check the numbering. So numbering will start from this nitrogen, first, second, third, fourth, and fifth. This is our substituent ring. Whereas the oxazole, that is with oxygen and nitrogen, 5-membered ring is our parent ring. Okay. So see, so for parent ring, we have to give the name of the site. That is, the first and second number atoms will get the site A. So this is A, this is B, this is C, this is D, and this is E. So you can see the attachment is in between this fourth and third carbon atom with this side D. Okay. And the substituent ring is pyrazole. That is 5-membered ring with 2 nitrogen, which is on first and second position. That ring is pyrazole. So it becomes pyrazole, and it is attached with fourth and third side of our pyrazole. So 4,3. Now why it is 4,3? Why it is not 3,4? Because if you will see the numbering of this oxazole ring is in this manner. Okay, so if it is in this manner, so we have to also consider the numbering in that manner only. That's why it becomes 4,3, not 3,4. And the site with which this oxazole is attached, that site is D. That's why we have to write D over here. So 4,3D and oxazole. Okay. So which option is similar with our pyrazole 4,3D oxazole? That is option B. Okay. Now next question. Identify the structure of 3-acetyl 4-chloro 6-ethyl 1H indole. Now what is indole? Indole is the fused compound in which the benzene and the 5-membered with nitrogen, that is the pyrrole ring, is fused with each other. Okay, so you can see this is having benzene with pyrrole, benzene with pyrrole, benzene with pyrazole, because it is having 2 nitrogens on first and second. Okay, it is not indole because there is no unsaturation over here. So the answer will be either compound 1 or compounds 2. As you can see, 3-acetyl, it means 1, 2, and 3 on third position. We are having the C double bond O with oxygen with methyl. And over here, C double bond O CH3. So I hope all of you know that acetyl is nothing but the C double bond O CH3, not this C double bond O with OCH3. This is the ester group. Okay, that's why the correct answer is compound 2, because it is having acetyl on third position, it is having chlorine on fourth position, it is having ethyl on sixth position. Okay, so the correct answer is compound 2, which is matching with this IUPAC nomenclature. Next question. Which of the following resonating structure of thiazole is incorrect? They are asking for incorrect resonating structure. Okay, so in case of thiazole, thia, it means sulfur, and aza, it means nitrogen, and ol, it means 5-membered ring. So this is thiazole. the sulfur is having lone pairs. Okay. So this lone pair will get shifted over here. When this lone pair will get shifted on this double bond gets shifted on this nitrogen. So we will get this nitrogen which is with negative charge. Okay. And the sulfur will get the positive charge because this sulfur is having 3 bonds. When this negative charge will get shifted over here, this bond will get shifted over here. Then we will get this nitrogen double bond over here. Here we will get negative charge. The sulfur with positive charge. When this gets shifted over here, this bond gets shifted on this carbon atom. This nitrogen is with this sulfur. Here double bond. Here negative charge. Double bond over here. When we will shift over here, this will get shifted over here. After this, we will get negative charge on this carbon atom. It is with double bond. This is with sulfur. This sulfur is having still positive charge. So you can see in every structure we are having sulfur with positive charge. But the second structure is with sulfur with negative charge, which is the incorrect resonating structure of this thiazole. Okay, so the answer is structure 2. It is incorrect resonating structure of thiazole. Okay. The next question is, which of the following 5-membered heterocyclic ring, which is most basic? They are asking for imidazole, pyrazole, thiazole, and oxazole. Now, in case of imidazole, we are having 2 nitrogens which are on first and third position. Okay. In case of pyrazole, we are having nitrogen. It is on first and second position. Okay. In case of thiazole, we are having nitrogen and sulfur on first and third position. Whereas in case of oxazole, we are having oxygen and nitrogen, which is on first and third position. Okay. Now see, in the case of imidazole, we are having 2 nitrogens on first and third position. Out of that, this nitrogen is having the lone pair, and that lone pair which will get protonated, that is not the part of pi system. Okay, that's why the basicity of this imidazole is highest as compared to other. Another thing, the pKa value of this imidazole is at around 7.1, whereas this pyrazole is at around 2.48, this thiazole is at around 2.5, and this oxazole is 0.8. We know that the pKa is directly proportional to basicity and inversely proportional to the acidity. Okay, so this is having the highest pKa. It means most basic because it is having direct proportionality. So the answer is option A. Okay, whereas the least basic is this oxazole. Okay. The next question is, which heterocycle is synthesized from malonic ester and urea? So see, in case of malonic ester, we will see the structure of that malonic ester. So malonic ester is having 2 ester groups in the same molecule. Okay. And urea. Now what is urea? Urea is NH2, C double bond O, NH2. Okay. So first thing you have to see whether the given compound is having 2 nitrogens in that structure or not. So in case of pyridine, it is having only one nitrogen. So this option will get cancelled. In case of imidazole, 2 nitrogens, 5-membered ring. In case of pyrimidine, 2 nitrogen, 6-membered ring. Whereas in case of indole, only 1 nitrogen is there. So this option will get cancelled. Now see, when we will react this malonic ester with this urea, we will get the 6-membered. How we will get the 6-membered compound? If we are considering this carbon, this carbon, this carbon, this 3 carbon atoms with this nitrogen, that becomes the 4th atom, this 5th atom, and this one 6th atom. So we will get this heterocycle. This heterocycle after the reaction of malonic ester and urea, and that is nothing but the pyrimidine derivative. That's why the answer is option C, pyrimidine. Next question, Pal-law synthesis of furan is. So you have to remember, whenever we are having Pal-law synthesis, so in that case, we always use 1,4 diketone. Okay, so if suppose I'm taking this generally 1,4 diketone, and if I want this furan, so I have to react this 1,4 diketone under the acidic condition. So when I'll react in acidic condition, I'll get furan, the substituted furan over here. Remember the another thing, when I react with this with ammonia, so we will get the pyrrole. And when we will react with this sulfur containing reagent, we will get the thiophene instead of furan. That's why the answer is cyclization of 1,4 diketone under acidic condition, that is option A. Next question, what is Chichi-Bibin reaction? Now see, this Chichi-Bibin reaction is the method. This is the synthesis method for the synthesizing of 2-aminopyridine. Now what is pyridine? Pyridine is 6-membered ring with 1 nitrogen atom, that is pyridine. And 2-amino, it means on second position there will be amino. But how we will get? So when we will react the pyridine with the sodium amide. Okay. So when we will react the pyridine with the sodium amide, that is NaNH2. Okay. So that nucleophilic substitution will occur on the second position only, because this nitrogen is having more electronegativity. It will pull the electrons towards itself. So this carbon will be having electron deficient carbon. That's why the nucleophile will attack on that position only, and we will get the 2-aminopyridine. So the answer is conversion of pyridine to 2-aminopyridine, that is option C. Okay, next question is bromination of thiophene gives. So thiophene is 5-membered with 1 sulfur. Okay, so when we will react this thiophene, okay, and when we are doing the bromination, after bromination, we will get this dibromo substituted thiophene. Okay, so the numbering will be in this manner. So 2,5 dibromothiophene will be our product after bromination. So the answer is option A, 2,4, 2,5 dibromothiophene. That is option A. Next question, identify the product of the following reaction. So here we are reacting this acetophenone with this hydrazine in presence of sodium hydroxide. Okay, so see, or sodium ethoxide. So which reaction is this? If you remember, whenever we react aldehyde or ketone with hydrazine and hydroxide, that is nothing but the Wolff-Kishner reduction. Okay, so when we will react with this hydrazine, first we will get, so see, this oxygen will combine with this hydrogen and it will remove out as water molecule. Okay, so instead of C double bond O, we will get C double bond N, and that nitrogen is with NH2. And the CH3 will remain as it is. So first we will get this. When we will react with the sodium hydroxide or sodium ethoxide, what we will get? So this will removed out and we will get simple hydrocarbon over there. So from this acetophenone, we will get this product. This product is ethyl substitution with this benzene. So the answer is ethyl benzene, not toluene, because it is not with methyl. It is with ethyl. Okay. So the answer is option B, ethyl benzene. Next question. Which of the following drug is used as anti-lipidemic agent? So the given options are, first is ranitidine. We know that ranitidine is used as antacid. Whereas the second option is celecoxib, which is used as NSAID in, okay. Atorvastatin is anti-lipidemic drug and the zidovudine is used in the treatment of HIV. So this is used in the treatment of HIV. The celecoxib is used as NSAID. Okay. Whereas this ranitidine is antacid. Okay. So the correct answer is option C, that is atorvastatin, which is used as anti-lipidemic agent. Okay. The last MCQ is identify the heterocycle in the given molecule. Now this molecule is having this heterocycle which is 5-membered with 1 oxygen. So the IUPAC nomenclature is oxole because 5-membered with oxygen. But the common name is furan. Furan is 5-membered ring with 1 oxygen atom. Okay. Now we will see some subjective questions. Now before that, I have to share one thing with you all. The Pharmacad has this course which include the video lectures of all the subjects. This course is specifically for semester 4. It includes the video lectures of organic chemistry, medicinal chemistry, pharmaceutics, ecology, as well as cognosy. Even this course has the model question papers with the solution so that you will get the idea about how to solve the question papers and which type of question they will ask in the exam. This course is in only 49 rupees. So if you want to purchase this course, you can contact to the given contact number. Okay. Now we will start. Now we will start with the subjective questions. Okay. So in subjective question, there are chances of getting resonating structures of different heterocycles, or you have to identify the configurations of the given compound, whether it will be geometrical isomerism or conformationals. Okay. Or there are chances of getting some definitions which you have to explain with the examples. okay, with these structures, because this is organic chemistry. So you have to explain everything with the help of structure of that particular compound. So we will see some subjective questions. So the first question is they're asking the resonating structures of pyrazole. So pyrazole, as we have discussed before some time, the pyrazole is having the 5-membered ring, and that pyrazole is with 2 nitrogens. Okay, and 2 nitrogens on first and second position. Okay, so this is pyrazole. Okay, so I'll tell you how to start the resonating structures. From which nitrogen you have to start, whether this one or this one. So see, the NH is having that lone pair which can delocalize, and because of that we have to start from this nitrogen. Okay, so this lone pair will get shifted over here. At that time, this pi bond will get shifted over there. Okay, so first we will get this with NH. When we are having this double bond over here, this nitrogen will get the positive charge. Okay, it is with negative. After this, this will get shifted over here, and this double bond will get shifted over here. Okay. So we will get negative charge on this nitrogen. We will be having double bond over here, and we are having double bond already on this nitrogen and carbon. After this gets shifted over here. This will get shifted over here. And you have to remember, you have to follow the resonating structures in only single direction. You can't change the direction in between. Okay. This bond gets shifted over here. So we will get negative charge on this carbon atom. After this, we will get this negative charge on this carbon and double bond on this carbon and carbon. We are having nitrogen with double bond, and this is with hydrogen. At the end, this gets shifted over here, and this double bond will get shifted on this nitrogen, and we will get this structure. Okay, so these are the resonating structures of pyrazole. So for this resonating structure, you should know that from which atom we have to start. Okay, and you have to follow the same manner. Okay, you can't change the direction in between. Okay, then the next is furan. So in case of furan, you have to draw in same manner. In case of furan, you have to delocalize the lone pair which is on oxygen. So in this manner, you have to start. Whereas in case of thiophene, you have to delocalize from this sulfur. Okay. The next question, which position of the imidazole ring is susceptible to electrophilic aromatic substitution? Now see, what is imidazole? Imidazole is 5-membered ring with with 1,3 nitrogens over there. Okay. Okay. This is imidazole. So see, this is first, second, third, fourth, and fifth position. So as we know that the nitrogen is more electronegative than the carbon atom. So this carbon will have the electron deficiency. So the nucleophile is able to attack over here because nucleophile is having electron with itself. But whenever we are having electrophile, it will not attack on the second carbon atom. It will either attack on fourth or fifth carbon atom. Okay, which one? Electrophile. So remember this thing. Electrophilic aromatic substitution at C4 or C5 because that both the carbons are electron rich carbon as compared to C2. Okay, C2 is having electron deficiency, so electrophile can't attack over there, only nucleophile is able to attack over there. After that, the reactivity order of this pyrrole, furan and thiophene. Now in case of pyrrole, we are having 5-membered with 1 nitrogen. The furan is 5-membered with 1 oxygen and the thiophene is 5-membered with 1 sulfur. So see, pyrrole has the trivalent nitrogen atom, and because of this trivalent nitrogen atom, it has electron rich carbon that is more reactive and it is less stable. Whereas in case of furan, which is having oxygen, it is less reactive as compared to pyrrole because of the electronegativity of sulfur. And it has oxygen and nitrogen. Okay, so it is having nitrogen, it is having oxygen, and it is having sulfur. Correct? So the order is first pyrrole, which is more reactive as compared to oxygen, that is furan, as compared to sulfur, that is thiophene. Okay. Then the next is, second question is, define the term stereospecific and stereoselective. So stereospecific reaction, it means it will yield a single specific isomer, the specific single isomer. Whereas in case of stereoselective reactions, it will give the unequal mixtures of stereoisomers. And out of that unequal mixture, the one isomer is formed more rapidly as compared to another. So in case of stereospecific, we can take example of bromination of alkene. Okay, bromination of alkene. So when we will do the bromination, we will get either this or this. Okay, the specific product. Okay, but whenever we are having the reduction, if we will take example of reduction of this compound, okay, it is with methyl. Okay, and if we want to reduce this with the help of, suppose, platinum catalyst. So after reduction, we will get two compounds. This with this and this. So this will be our major, that is 68%, and this will form in 32%. So this is the example of stereoselective. We will get the selective product, the unequal mixture. But in this case, we will get the equal mixture, the 50% this and 50% this. So that is nothing but the racemic mixture. So remember this thing. Whenever there is stereospecific reaction, we will get the racemic mixture. And whenever there is stereoselective reaction, we will not get the racemic mixture. There will be unequal mixture. Okay. Then they're asking mechanism of halogenation of cis-2-butene and trans-2-butene. So see, if we are having cis-2-butene, it means this. And we are adding Br2. Okay. So what will happen? This lone pair will get shifted on this carbon atom, and this double bond will get shifted on this bromine, and this bond will get shifted on this bromine. So after the reaction, we will get this methyl with this methyl. This bromine will be having positive charge, and the second bromine will be with negative charge, which will attack from this side. This bond will get shifted over here. So at the end, we will get this product. Bromine on this side, bromine on this side. This is a kind of anti-addition of bromine. Okay, but when we are having the trans butene, that is in this manner, and we are adding the Br2 over here, okay, what will happen? Shifting over here, this will get shifted over here, and this will get shifted on this bromine. After this methyl is on this position, dash wage, and the another methyl is on solid wage because it is at the another site. Okay, this bromine which has positive charge, and this bromine which is with negative charge, this will attack from this side. Okay, and we will get this product. Methyl with solid wage, with dash wage, and this bromine. So this is the difference in between when we are reacting with cis-butene and when we are reacting with trans-butene. And they are asking whether the halogenation is stereoselective or specific. We have already discussed it is stereospecific reaction. Okay, I hope these two questions are clear to all of you. We will see next question. So they are asking resolution of racemic modification. So what is resolution? See, resolution is nothing but the process with which we are able to separate the racemic mixture. And we know that the racemic mixture is having 50% one configuration with 50% another. If it is dextro, the another one is levo. And we will get the racemic mixture which is optically inactive. If we want to separate this dextro and levo from this racemic mixture, we have to use the techniques of racemic modification. So it has different technique. The first technique is mechanical method. So mechanical method is by using the crystallization. So in this method, what we can do, first we will crystallize this whole racemic mixture. See, after crystallization, because of the different nature, they will be having the different shapes of crystals. So with the help of magnifying lens and forceps, we are able to separate. Okay, and the basis of the shape of the crystal, we can identify whether it is dextro or levo. But it is very time consuming technique. The second technique is biochemical separation. So in biochemical separation, what they usually do, see, certain bacteria, fungi or yeast grown in dilute solution of this racemic mixture. So that bacteria or fungi will eat up out of these two, one enantiomer will get rapidly destroyed by this particular bacteria. Okay, example, Penicillium glacium is there, which destroys the dextroisomer of ammonia tartary. So that Penicillium glacium will destroy the dextro enantiomer. So at the end, we are able to separate the levo. But this separation technique is having one limitation because one enantiomer will get destroyed out of this two. Okay. The third method is chromatographic method. So chromatographic method is on the based on the separation on the basis of optically active support. Okay, so in which we can use the diastereomeric adsorbents. So the different enantiomer is having the different stability on that particular absorbance. Okay, and different enantiomer will different held tightly differently with that particular adsorbent. Okay, and those who is loosely bind with the adsorbate, that will elute first. So on the basis of elution, we are able to separate the dextro and levo enantiomer from the racemic mixture. Okay. The last method is kinetic method. So what we usually do, we will react this racemic mixture with the different reagent. So suppose we are reacting the mandelic acid, racemic mixture of mandelic acid with the menthol. So the dextromandelic acid will react faster as compared to levo. Okay, so in that basis, we are able to separate it out, the dextro and lev. Okay, so racemic modification has total 4 techniques: mechanical, that is crystallization, the biochemical, the chromatographic method, and the kinetic method. Okay, next question is they are asking the mechanism for Van Leusen oxazole synthesis and Hans pyridine synthesis. So Van Leusen oxazole synthesis includes the synthesis with aldehyde, okay, aldehyde with tosyl methyl isocyanide, okay, and in the presence of this potassium dichromate, we will get the oxazole. Okay, whereas in case of Hans pyridine synthesis, in this case we have to react the aldehyde with beta keto ester. Okay, so beta keto ester, so we have to take why it is beta. See, this is alpha carbon, this is beta carbon, so we have to take 2 moles of this beta keto ester. After reaction, we will get the substituted pyridine. Substitution with this ester. Okay, so in case of Van Leusen oxazole, we have to use aldehyde with tosyl methyl isocyanide, and in case of Hans, we have to use the beta keto ester with the ammonium acetate. Okay. The next question, draw conformations of N-butane with energy profile diagram and the stability. So see, these are the conformations. So in this case, see, butane, it means what? Total 4 carbon atoms. So if we are considering this as the frontal and this as the rarer one, the round one, so this carbon is with 2 hydrogen and 1 methyl. This carbon is with 2 hydrogen and 1 methyl. So first, when we will rotate this with 60, we will get the gauche. This is eclipsed because the angle in between 2 carbons is 0. Okay. When it becomes 60, it becomes eclipsed. Eclipsed to staggered. So this is eclipsed. This is fully eclipsed. This is partial staggered because 2 bulky groups are nearer by each other. After that, when we will rotate this with 60 degree, we will get this eclipsed. After the 60 degree rotation, we will get this conformation, that is the partial staggered. After 60 degree rotation, we will get the partial eclipse. And after 60 degree rotation, we will get the staggered one. And this is fully staggered because 2 bulky groups are on opposite side. This is the energy profile diagram. So this fully eclipsed is required this much energy, whereas this partial eclipse requires 3.6. this eclipsed will require again 3.6, and this eclipse required 5 kilocalorie per mole energy. So on that basis, we can conclude the stability order of all of these conformers. Okay, so we know that energy is inversely proportional to the stability. Okay, now we will see the next question. What will be the product of Birch reduction of benzoic acid? So what is Birch reduction? Birch reduction is nothing but when we will add sodium in presence of liquid ammonia. So we are having benzoic acid. Benzoic acid, it means benzene with the electron withdrawing group. So remember this thing, whenever we are having benzene with electron withdrawing group and we are doing the Birch reduction, the reduction will take place on the ipso position. Okay, it means over here and on para position. So we will get this product. Okay, remember when we are having electron withdrawing group, that is our benzoic acid. But when we are having electron donating group instead of withdrawing, so after reduction, we will get the reduction at which position? On para position and on meta position. Okay, remember, withdrawing group, we will get protonation on ipso position and para position. Donating group, we will get reduction at para position and meta position. Okay.
Ma'am, your mic is off.
Whenever we are adding this bromine with oleum, with this pyridine, okay? So when we will add this, what we will get on pyridine, the bromination will occur on which position? Okay, so it will occur on this third position. Remember this thing. Okay, when we are doing nitration of this thiophene in presence of this acetic anhydride. Okay, so what will happen when we are doing nitration in case of thiophene? So there are chances of getting substitution on the second position or on the fifth position. Okay, and out of this, the major product will be this one. This is the major product. Okay, when we are adding MCPBA, what is MCPBA? It is metachloroperoxybenzoic acid. So when we are adding this on this pyrimidine, so pyrimidine is 6-membered with 2 nitrogen on first and third position. Okay, so when we will add MCPBA over here, we will get the oxygen on this nitrogen. So we will get N-oxide pyrimidine. And this nitrogen will be having positive charge. Okay, so these types of question they may ask in the exams. They will ask the different chemical reactions as well as the synthesis methods of heterocycles. Okay, so the last question is using various oxidizing and reducing agent, discuss the oxidation and reduction of isoquinoline. So what is isoquinoline? Isoquinoline is with benzene and it is with pyridine, but that nitrogen is on second position. Iso, it means on second position. Okay, 1, 2, 3, 4, 5, 6, 7, and 8. So see, if we are using the KMNO4, but if it is neutral KMNO4, okay, so when we are using the neutral KMNO4, we will get this phthalimide. Remember this thing. But when we are using the acid alkaline KMNO4, okay, alkaline KMNO4 whenever we are using, we will get the phthalic acid. Phthalic acid from the benzene ring, okay, and plus we will get this pyridine with this 2 carboxylic acid group. Okay, that is pyridine 4,5 dicarboxylic acid. Okay, so when we are using neutral KMNO4, we will get phthalimide. But when we are using alkaline KMNO4, we will get this phthalic acid and this pyridine dicarboxylic acid. Like that, when we are using the hydrogen platinum as a reducing agent, okay, so when we are using the platinum catalyst as a reducing agent, the reduction will happen on all the positions. Okay, remember this. But when we are using the diethyl aluminium hydride, that is C2H5 twice with the aluminium hydride. So there will be reduction only on first and second position. Remember this thing. The remaining position will remain unsaturated. Okay. So we will get 1,2 dihydroisoquinone. Okay. So on the basis of reagent, our product will get different. Okay. So that's all about the paper solving session for POC 3. If you have any doubt, you can add in the comment box. So the Pharmacad has this course which has the video lectures of all the subjects, which includes organic chemistry, medicinal chemistry, physical pharmaceutics, pharmacology, cognosy, as well as it is having the model question papers with the solutions, so that you will get the idea which type of questions will ask in the exam and how to solve those questions. This whole course is for semester 4 only in 49 rupees. So if you want to purchase it, you can contact on this given contact number. Thank you.